The aim of this procedure is to manufacture and test cellular uptake, secretion, and antiretroviral properties of crystalline antiretroviral nanoparticles, which we refer to as nanoart. This is accomplished by first processing the antiretroviral drugs by sonication, homogenization, or wet milling. The second step of the procedure is to characterize the physical properties of the nanoart particles. After primary monocytes are isolated by countercurrent centrifugal elutriation, they are cultured for 7 days in DMEM containing macrophage colony stimulating factor, or MCSF, which induces their differentiation into monocyte-derived macrophages. The third step is to test the uptake, retention, and release of nanoart in human monocyte-derived macrophages. The final step of the procedure is to test the in vitro antiretroviral efficacy of nanoart in monocyte-derived macrophages. Ultimately, results can be obtained that show the facilitated uptake and slow release of nanoart from cells and robust antiretroviral efficacy. These are determined by measurements of drug levels by high-performance liquid chromatography, or HPLC, and by testing infected culture supernatant materials by measurement of reverse transcriptase activity.
Hi, I'm Shantanu Balkundi, and I'm a postdoctoral research fellow in Dr. Howard Gelman's lab at the University of Nebraska Medical Center. The methods we present can help answer key questions in nanomedicine that include how cell-based drug delivery systems can be used to develop translational research activities. This can be accomplished by using nanoformulated antiretroviral therapy with prolonged half-lives. Our studies support the potential use of nanoart for clinical use.
Hi, my name is Ari Nowacek, and I'm an MD PhD student scholar working in the Carol Swartz Emerging Neuroscience Laboratory under the guidance of Dr. Gendelman. The implication of the techniques that we show you here extend to antiretroviral therapy because nanoformulated compounds can simultaneously improve bioavailability and pharmacokinetics while also reducing drug-related side effects. By taking clinically available antiretroviral medications and repackaging them into nanoparticles, we may be able to improve compliance and also positively affect disease outcomes.
Hi, I'm Dr. Howard Gendelman. I'm a clinician scientist and professor and chair of the Department of Pharmacology and Experimental Neuroscience here at the University of Nebraska Medical Center. Our aim overall is to simplify antiretroviral drugs, to make them easily available and improve the biodistribution and pharmacokinetics. Now I turn you over to Ari and Sean, who will bring you into the research laboratory and show you how the manufacture and development of these nanoparticles are done, clinical utility and development here at the University of Nebraska Medical Center.
To manufacture nanoart via wet milling, first weigh the drug crystals and various surfactants that will be used to make the nanoformulations and mix them with 10 millimolar Heapes buffer, pH 7.8, using a T18 Ultra Turex mixer until completely dispersed. The percent of the drug in the formulation should be between 1 and 2%. For continuous milling, make sure that the chiller and the compressor are on, and that the outlet pressure is around 100 psi. Now set up so that the grinding media can be loaded in the tank. And add 50 milliliters of beads to the grinding chamber using a funnel. Make sure that there are no beads in the threads or anywhere outside the grinding chamber to avoid leaks in the mechanical seal. Now make sure that the O-ring is in its place on the milling chamber. Then, select an appropriate screen mesh size and secure the lids by tightening the nuts. The screen mesh size should be at least half the size of the beads. Use a large screen mesh size to avoid the product from clogging the filter while continuous milling. After lowering the grinding chamber, add the drug suspension with various amounts of surfactants to the product inlet. The minimum volume of the suspension should be 100 milliliters. This will prevent the milling chamber from overheating and also avoid bead contamination due to less wear on parts. Next, start the pump using the control unit. varying the flow rate as required for your formulation. Then, turn on the agitator and adjust the speed on the control unit. The speed can be increased from 600 rotations per minute to 4,320 rotations per minute on a micro SIR. Monitor the temperature using the temperature gauge that is attached on top of the grinding chamber to make sure that there is no overheating. Now mill the sample at various speeds and flow rates and take out small aliquots of about 1 milliliter of the formulation for size and charge measurements using a diffraction light scattering Malvern zetasizer. After the desired size is obtained, stop the milling using the control unit and with the pump still on, collect the sample into a flask, allowing the drug to drain completely. Remember to switch off the pump when done and remove and clean the beads. Now transfer the milled suspension into 50 milliliter centrifuge tubes and set the centrifuge to 10,000 rotations per minute for 30 minutes. Upon completion of the centrifuge cycle, measure the amount of supernatant and replace with the same amount of fresh surfactant solution. Once resuspension is complete, transfer the suspension to the milling chamber and mill for 10 minutes. Take an aliquot of about 1 milliliter and measure the size and charge of the sample, again using the zetasizer. Finally, measure the concentration of the nanoart formulations using HPLC. To manufacture nanoart via homogenization using the Avastin Emulsiflex C5, prepare the drug crystals and various surfactants as previously described and transfer the suspension to the homogenizing vessel and begin recirculation. Make sure that the chiller is on before starting to homogenize the sample. Increase the pressure gradually until the meter reads 20,000 plus or minus 2,000 psi and continue to homogenize until the target particle size is achieved. This usually takes between 45 to 60 minutes. Take an aliquot of about 1 milliliter periodically and measure the size and charge of the sample using a zetasizer. Centrifuge the homogenized suspension as previously described. Then, after resuspension, transfer the suspension to the C5 homogenizer. Restart the circulation and return the homogenizing pressure to 20,000 plus or minus 2,000 psi and homogenize for 30 minutes. To manufacture via sonication using the Cole-Palmer ultrasonic processor, pour the dichloromethane, PLGA, and drug solution into a 1% PVA surfactant solution and place in an ice bath in the ultrasonicator. Start sonication at 50% amplitude, although the amplitude of the sonicator can be reduced to about 30% amplitude for smaller batches of samples. Sonicate the sample for 10 minutes. Take out a small aliquot for particle size analysis. If the size of the particles is greater than 1.5 microns, sonicate the samples at 2-minute intervals up to a maximum of 16 minutes total. Observe the samples under the microscope at 20 times magnification and document observations. Use a stir plate to create an adequate vortex for the remaining suspension and continue mixing overnight at room temperature. Collect the suspension after 24 hours and pour into 50 milliliter centrifuge tubes. Centrifuge the samples at a speed of 8,000 rotations per minute for 20 minutes at 5 degrees Celsius. Decant the supernatant and resuspend one half of the sample in 75 milliliters of filtered reverse osmosis water and the other half in 75 milliliters of 0.5% cetyl trimethyl ammonium bromide surfactant. Centrifuge the samples again at a speed of 8,000 rotations per minute for 20 minutes at 5 degrees Celsius and resuspend each of the pellets with a total of 20 milliliters of mannitol solution. Finally, for particles manufactured via either homogenization or sonication, measure the size of the particles using the zetasizer and measure the concentration of the samples using HPLC. To prepare for visualization with microscopy, take the prepared nanoart suspensions and sonicate them for 10 seconds at 20% amplitude with a sonication probe. Take a microscope slide cover slip and place it on an inverted microscope. On the cover slip, mix 15 microliters of the nanoart suspension with 100 microliters of phosphate buffered saline and mix by pipetting up and down several times. Then, visualize the nanoparticles using a 20 times objective. To visualize morphology, samples can also be prepared for visualization with scanning electron microscopy. Here, we see images obtained from a JEOL JSM 6300F low voltage field emission scanning electron microscope. After isolation of blood-borne monocytes by centrifugal elutriation, add purified monocytes at a concentration of 1 × 10⁶ cells per milliliter to DMEM supplemented with the following additives. Then, plate 2 × 10⁶ cells per well in 6 well plates within a humidified atmosphere supplemented with 5% carbon dioxide. And culture for 7 days in order to allow monocytes to differentiate into macrophages. To analyze cell uptake and collection, mix nanoart with culture media to a final concentration of 100 micromolar and add 1 milliliter of treatment medium to each well. At the desired time point, or when cells are fully loaded with nanoart, remove all treatment medium and wash the cells 3 times with 1 milliliter of PBS to remove any particles that have not been taken in by the cells. In 1 milliliter of PBS, remove adherent cells from the bottom of the well by using a cell lifter and place them into a 1.7 milliliter microcentrifuge tube. Now centrifuge the cells at 1000 RCF at 4 degrees Celsius for 10 minutes. Remove the supernatant and add 200 microliters of 100% methanol. Lyse the cells and dissolve nanoart by briefly sonicating the pellet with a sonicating probe at 20% amplitude for 2 to 5 seconds. Store the samples in -80 degrees Celsius until ready to analyze drug content. The intracellular retention of nanoart can also be visualized using transmission electron microscopy after sections are prepared via microtome, as seen here. See the accompanying text to this article for details. To analyze nanoart release, treat cells as previously described for analyzing cell uptake and collection. However, instead of collecting cells after washing, replace PBS with 2 milliliters of cell culture medium without MCSF and without nanoart. On indicated days, or as indicated by the media turning yellow, exchange half of the medium. At the desired day, collect 1 milliliter of the cell medium along with replicate cells and process for drug analysis. To visualize real-time uptake of nanoart by confocal microscopy, first take a dye such as Vibrant and add it to the medium following the manufacturer's instructions. Add the dye containing medium to the mature monocyte-derived macrophages and then rinse the cells 3 times using 1 milliliter of culture medium to remove any excess dye. Now mix nanoart with culture medium using fluorescently labeled nanoart. And add treatment medium with labeled nanoart immediately prior to imaging with a confocal microscope. Capture an image every 30 seconds for 4 hours using a 60 times objective. Finally, HIV-1 infection and infectivity assays can be performed. First, treat cells as described for visualizing art release. Then, on desired days, remove all medium and replace with medium without MCSF containing HIV-1 ADA at a multiplicity of infection of 0.01 viral particles per cell and incubate for 24 hours. Then, remove the viral medium and replace with fresh, virus-free media. Culture cells for 10 days, exchanging half the medium every other day or as necessary to keep the cells alive. 10 days post-infection, collect 10 microliters of medium from each well, place into a 96 well plate and store at -80 degrees Celsius until ready to perform reverse transcriptase analysis. Subsequent HIV-1P24 detections can also be performed. Here we can readily see the morphology of cells exposed to virus at a multiplicity of infection of 0.01 one day after nanoart treatment. In these monocyte-derived macrophages, there is no evidence of viral infection, and the nanoparticles are readily visible within the cell's cytoplasm. Here, cells were exposed to virus 10 days after nanoart treatment. Some of these cells were infected, as indicated by P24 staining and multinucleated giant cell formation, but there was still significant protection as compared to untreated cells. Note that nanoparticles are still visible within the cytoplasm of cells in B, but not as much as was seen in A. These structures are not present in cells that were not treated with nanoart. In these live cell confocal videos, we see macrophages labelled in green and nanoart labelled in red. Here we can see that the cells accumulate very few nanoparticles. Here they rapidly accumulate particles as indicated by increased red fluorescence. These differences parallel the findings that are seen when analysing drug uptake using HPLC. In addition, we can see the cells actively take up the particles. The difference between rates of cellular nanoart accumulation reflects the effects of physical characteristics such as size, shape, and charge on the abilities of macrophages to uptake and retain nanoart.
After watching this video, you should have a good understanding of how to manufacture nanoart using wet milling, homogenization, and sonication techniques. These are effective methods to manufacture antiretroviral nanoparticles. Wet milling and homogenization serve to fractionate larger drug crystal particles into smaller nanoparticles within lipid surfactants, and a sonication technique produces individual nano-sized drug droplets within a polymer.
Here we've also demonstrated how to use human monocyte-derived macrophages as a testing platform to assess nanoart transport. Our method for testing allows for a very simple screening of a particle's ability to be taken up, retained, and released by cells, as well as their capacity to inhibit viral replication. This allows the researcher to identify nanoart that may have the best chance of succeeding in vivo and ultimately for use in infected humans. Complete results of these experiments are published in our article online. The article is named Nanoformulated Antiretroviral Drug Combinations Extend Drug Release and Antiretroviral Responses in HIV-1 Infected Macrophages, Implications for NeuroAIDS Therapeutics, in the Journal of Neuroimmune Pharmacology, 2010. Thank you very much.